So the first step of the HIV life cycle is attachment. The key players here on the HIV virus are the GP41 and GP120 proteins encoded by the N genes. Here in the human host cell, we have the CD4 receptors and two co-receptors called CXCR4 and CCR5. The GP120 proteins attach onto the CD4 receptor as well as the co-receptors.
In the next step, the HIV has to enter the human cell. This involves an HIV protein called GP41 encoded by the gene ENV. Here, the membranes of the human cell and the HIV cell start to fuse together. Reverse transcriptase, another viral protein encoded by the Pol gene, first takes the single-stranded viral RNA and makes its complementary DNA strand. Next, the RNA is erased and a secondary complementary DNA strand is created.
The next step is the integration of the provirus. The viral enzyme integrase splices the host DNA and inserts the viral DNA into the host genome. Now, as a provirus, it can lay dormant until all of the right transcription factors are present. Integrase is also encoded by the Pol gene.
First, the DNA is transcribed into RNA using host and viral proteins, host, TAT, and Rev, encoded by the TAT gene. Next, the RNA is translated into protein using host proteins called host and Rev, encoded by the Rev gene. Next, the protein is shortened into mature proteins using a viral protein called protease, encoded by the Pol gene. Next, the genome is packaged by the viral protein called GAG, which is encoded by the GAG gene. Then, the virion is ready to exit the cell. This involves viral proteins GP120 and GP41, encoded by the ENV gene. Host proteins are likely also involved.